Так, ну все, считаем, что кворум есть. 18:01. Коллеги, всем добрый вечер. Значит, с большим удовольствием представляю нашего сегодняшнего докладчика, выпускника нашей BayesGroup, моего хорошего друга Мишу Фигурнова, который уехал работать ученым, исследователем DeepMind. И, соответственно, в прошлом году, по-моему, да, в прошлом году, значит, коллектив исследователей DeepMind, включая Мишу, опубликовали статью в Nature про применение методов машинного обучения для предсказания критичной, может быть, даже четвертичной структуры белков, алгоритм, который назвали AlphaFold, и Миша любезно согласился нам про него рассказать. Миша, прошу вас.
Спасибо большое, Дмитрий Петрович. Очень приятно снова выступить на спецсеминаре, родном спецсеминаре, так сказать. Значит, технический нюанс. Я не очень хорошо умею пользоваться Zoom, так что если там кто-то что-то будет поднимать руку или говорить в чат, вы мне, пожалуйста, скажите, я могу это просто не увидеть. Так, okay, I will probably speak in English because I don't know all the words there are to describe the protein structure prediction. So I'll switch to English now. Okay, so I will present the work of this great group of people, and yeah, it's been a great pleasure to work on them with them on AlphaFold. So the outline of this talk will be as follows. I will first talk a little bit about what is the protein structure, and then I will describe how AlphaFold works, and talk a little bit about how AlphaFold understands proteins, then discuss the results of AlphaFold, and talk about what is next for machine learning in proteins and in protein structure prediction. Okay, introduction to protein structure. So what is a protein? It is a long molecule or chain of amino acids, and they are chained together with this peptide bonds. So it's like, it basically looks like this. So each of these segments is called an amino acid, and they differ from each other. There are 20 different types of amino acids. So if you're a computer scientist, you can think of this as a alphabet of 20 letters. And the letters are called either residues or amino acids. Basically, the residues are these bits which hang on the sides and which actually differ between different amino acids, but people refer to different amino acids as residues too. And each of these 20 letters is designated with a specific atoms and how they are bonded. So basically, there are 20 types of chemically different amino acids which are typically used. And if you have a DNA sequence, then this will directly encode the protein sequence. So if you are able to read the DNA, then you know which protein will be synthesized from that piece of DNA. Great. Any questions about what is a protein? Okay, let's move on. Here are the 20 different amino acids. And yeah, you can see that all of them are composed of a very small number of distinct atom types. Like, of course, the main of them is carbon, which is not even shown there. Like, those dots are the carbon atoms, but also nitrogen, hydrogen, oxygen. You can also find a sulfur here. So yeah, that's basic. They're very simple in terms of the atom types which are used there. But they differ from each other in terms of their properties. So some of them will be positively charged, others will be negatively charged. Some of them are polar, which means that they like to interact with water, and others are hydrophobic, which means that they want to hide away from water. And essentially, that's what makes them useful. Like, there is a wide range of amino acids, and they exhibit different behavior depending on the type. And as one example, here are the, this is basically an example of a protein which has been stretched into a long chain. And then you have the backbone here in the center. And there are the side chains hanging on the sides. And some of them are of these side chains will be hydrophobic, the ones on the left, and the others will be polar, which are the ones on the right. And so you can expect that the ones on the left will basically want to form the core of the protein, so hide away from the water, and the polar ones will form the surface of the protein, which will be exposed to water.
Michael, I have one question about the backbone.
Sure.
All the connections within the backbone are the same, of the same size?
Yes, it's those are the, well, there are three atoms: nitrogen, carbon, carbon, right? And then there is the peptide bond between, yeah, like all of them are covalent bonds, so it's very, very strong bond. And
okay, thank you.
it basically iterates like nitrogen, carbon, carbon, nitrogen, carbon, carbon, etc. Yeah. Right. So that's basically how it works. And then the amazing fact about proteins is that if you just put them into water, they fold in these extremely intricate 3D structures. And it's basically just, in most cases, it's just the consequence of physics. And if you look at the 3D structure of a protein, like on the left, it looks a bit like a, just random mass of atoms. Like you can't see too much there. So people often visualize it in a different way using the secondary structure as in the center. So it's just the backbone atoms, and like there is a cartoonish way to show some common patterns which occur in the backbone of the protein. And then it looks like this, and you immediately see that there is quite intricate structure in that. And maybe the most important fact for what I will be talking about today is that there are lots of interactions between residues which are far away in the sequence. So you can have like residue 10 in contact with the residue 1000 or something like that. So they can be quite far apart in the sequence, but very close together in the 3D space. Yeah, and on the right, you just see how these two align together. And people visualize either the cartoon version or the whole atom, depending on what exactly they want to understand about the protein. Because it's very important also to see the individual, the small details of the protein, because that's what typically does the actual chemistry of the protein. Okay, any questions about this? Okay, let's move on. So as you probably know, proteins are the machines of the cell, and that is why they are so interesting. And as a particular example, this is I think this is calcium pump, which is used in your muscles in order to relax them. So this is the machine which basically pumps calcium in some direction. I don't remember which one, between the muscles and the surrounding area. And amazingly, this is like literally one protein, and it takes in ATP, which is like the fuel of the of cells, and uses it to pump calcium atoms. And if you think about this, this is just incredible, because it literally can identify these calcium atoms and just pump it just that and in just one direction. So those are absolutely mind-blowing in my opinion, as a computer scientist who basically is not used to this sort of precision at this extremely small scale. And this video is taken from PDB 101, which is a really good resource if you want to learn more about proteins. Yeah, so proteins are basically what does all the chemistry in all living organisms. So they are quite important. Now, in order to understand the function of the protein, what they do, how they work, it's important to know their structure. But unfortunately, experimental protein structure determination is really hard to do. And to the huge credit of the protein community, they have a protein data bank which has recently marked its 50th year, where all the experimentally determined structures worldwide are being deposited. So it's a freely available data bank of the protein structures, and you can access them. And we used it for the AlphaFold training. And 200,000 structures does not sound like too much, but a single one of these structures can take years of a PhD student's work. It's not uncommon for a PhD student to basically just solve one structure during their entire PhD. So it's like a huge amount of work. to pull this off. Okay, any questions about this?
Just one more. Please turn off your mics.
Okay.
There are noise from someone.
Yeah. So again, as a computer scientist, it was really interesting for me to learn about the different ways to determine the protein structure. I don't have time to talk about this today, but yeah, it's a very, very cool topic. Yeah, just to give you an idea, you basically can't see these proteins because they're way too small. So if you just shine the light through them, they're completely transparent. Just to, to give a sense that you can't just look in a microscope and see them. Cool. Now I will talk about how AlphaFold works, and I will start with a disclaimer that I won't be able to talk about all of it. We have the Nature paper which was released this summer, published this summer, and the title is the same as the title of this talk, which is Highly Accurate Protein Structure Prediction with AlphaFold. And it includes a very comprehensive description of AlphaFold. It has 60 pages of supplementary materials with 32 algorithms and lots of diagrams you can see on the right. Yeah, algorithms describing everything. And also there is the code, which we have open sourced, which allows the community to build upon our work. And you can find it on GitHub. And if you would just like to play around and fold your own protein, you can do this without installing anything. You can just click this link. It's a Colab where you basically enter the protein sequence you would like to fold. You wait for a little bit of time, and you get the prediction out. So it's very easy to use and to play around with. Yeah, so check it out. Check it out if you're interested. Okay, I will start with explaining basically the principle behind both AlphaFold and many other methods for protein structure prediction, which is evolutionary structure prediction. So what's the approach here? So we have this sequence of the protein, which we know this, which we want to figure out the structure of. And there is a causal direction from sequence to structure, which I basically talked about. Like, if you just throw it into water, you get this structure. But there is another dependence. Probably, if this protein exists, probably it does something, right? So it probably has some sort of function. And during the course of evolution, there is a constant evolutionary pressure. Like, you have random mutations, you have additions, deletions in your genomic code, which causes corresponding changes in the protein, right? So you have the evolutionary history. If you look at the evolutionary related proteins, these will be the ones which have mutated, but they still work. And you can assume that these proteins also have a very similar structure, maybe even almost the same. So you have this sort of glimpse into the world of possible proteins which fold, possible sequences which all fold in very similar structures. And if you look at that, you can go back in like a Bayesian way and form the problem of evolutionary structure prediction. So you augment your sequence with these evolutionary related sequences, and then you say, given that, can I figure out the structure of the protein? Does this make sense?
It does, yeah.
Okay. So now in slightly more detail, what people do is you start with a sequence, you search the genetic databases for this evolutionary related sequences, and it's called multiple sequence alignment. And the word alignment is quite interesting here because it actually uses a hidden Markov model in order to find the best possible alignment between all the sequences, which works surprisingly well. Like, you don't often see hidden Markov models being, at the core of something, and still, in this day and age where everything has been replaced with neural networks. But this actually works surprisingly well. And yeah, it basically is like the most likely alignment of all the sequences under some graphical model. And once you have that, you can look at this matrix and find some interesting properties. Like, for example, looking at just the single column, you can look, okay, if it's a hydrophobic residues most of the time, then probably it wants to be inside the protein. If it's hydrophilic, then probably it wants to be outside. And another useful signal is the coevolution, which is the fact that residues in contact often mutate together, which is not that obvious at the first sight. But if you think about it, imagine that you have two residues which are in contact, and one of them, let's say, is positively charged, and another is negatively charged. Okay, and then if one of them mutates, if both of them become positively charged, the protein will just break apart because they will no longer attract to each other, right? But if the order of them flips, so it's basically another one becomes negative and another one becomes positive, then probably the protein will still fold just fine, and the function will be preserved most of the time. As a result, you see a lot of correlation information in this multiple sequence alignments, which are also useful in determining the structure. Okay. Now let's talk about the actual, neural network ideas that we've used. And
Michael, I have one question.
Sure. Yeah.
What is the data source for this MCA algorithm? I didn't quite understand. What are the proteins you are trying to align?
Oh, so there are large genetics databases. For one of them is called UniRef. There are also metagenomics databases, and they have they differ by basically the quality of the data, but the good quality ones have hundreds of, like 100 million sequences, genetic sequences, and the low quality ones have like tens of billions. And the reason they exist is because it has become extremely cheap to figure out the sequence of from the DNA. Reading DNA has become extremely cheap. So people do that all the time, and they do all sorts of research on these databases. And we basically use these publicly available databases to search in those.
So do you understand, right, that first you decode the genome of different species, then you take the corresponding proteins, the chain-like structure of the protein, right? And then you're trying to run this multiple sequence alignment between different species.
Yeah, yes, that's correct. So it's a completely different species, and you basically just align the proteins between those. And generally, the rule of thumb is that if you can align two proteins between different species, probably they are doing roughly the same thing, or at least they are evolutionarily related. So that's basically the principle behind this.
And do you understand,
so basically, the space is so large you can't just have a good match between two sequences just by chance.
And do you understand, right, that actually for most of species, the most of proteins are related? So, for example, if we take human, we have how many? About 20,000 of different proteins, and the most of them are related with some proteins from the early steps of evolution, from different species, right?
Sorry, do you mean proteins like that? All the proteins within human are evolutionarily related, or
probably not all the proteins, but the most of them.
I think
related with the
yes, but here we're looking at the given protein. We try to find similar proteins in other organisms, but although if you find a similar protein in human as well, that's also useful, right? But most of the time they come from different organisms.
Okay, but what I meant is that actually there are very few proteins for a given organism that are not related with any other proteins from different species, because the most of proteins are very
that is correct. Yes, you can find those if you look at very, very rare or rather understudied types of organisms. Like if you look into somewhere very hot or something like that, or, some special cases of life, basically, then you may find those. But normally this just means you didn't look hard enough for other organisms there. Right?
Okay, thank you.
Yep. Okay. Right. Now let's talk about the inductive biases. And this is just like an overview slide saying that, yeah, inductive biases is important in deep learning, which is probably something you know already quite well. But yeah, basically, you can think of this as encoding the information flow into your network. And that means that some patterns become easier to find than others. But that is often very useful. So for example, with a convolutional network, which is what we used in the previous version of AlphaFold, you assume that your data forms a regular grid, and then you have information flow between local neighbors. So it's like a very strong locality bias. And you can already guess that maybe that's not the best fit for proteins, actually, because there is no information flow should not happen locally. Like you can have very non-local interactions. And then with recurrent networks, your data is assumed to be ordered sequentially. So information flow is sequential. People have also applied this to protein structure prediction. It kind of makes sense because you have a sequence of amino acids. But then again, as I said, you have non-local information flow, ideally. So yeah, there are graph networks, which are really good if you know the graph structure of your protein or your data. But normally, these graph networks assume knowledge of this graph structure. And as I will explain in like next few slides, that's basically what we want to find for a protein. And then there are attention models, which are assume that the data has unordered set, forms an unordered set, and use the information flow, which is determined by the network. And basically, with AlphaFold, we came up with essentially a new inductive bias for neural networks, which is much better suited for protein problems. So to summarize, our contribution here is that we have built our physical and geometric insights into the neural network structure rather than some process around this network, as has been sometimes previously done. So like the network is built in a way which allows it to learn this physically plausible information flows. And another contribution is that we have an end-to-end system which directly produces a structure, as opposed to, for example, predicting the distances, a distance matrix, and then somehow creating the structure out of that in a post-processing step. Now, when I say that we have inductive biases which reflect the knowledge of the physics and geometry, what do we mean by that? The first thing is that we specifically de-emphasize the position of residues in the sequence. So we pay as little attention as possible to the fact that this is a residue number 500 and this is residue number 550. We try to make the network as agnostic to that as possible. Of course, it still needs to know that, but we don't use some sort of a sequential processing or something like that, because we know that these residues could easily be in contact even if they are very far apart. And that means that the network iteratively learns a graph of the residues which are close by spatially, and iteratively tries to improve that graph as it's being built. So it tries to figure out whether the current graph structure actually makes sense. And we represent that graph as essentially a pairwise matrix of where you have residues here and the same residues here, and then the position ij gives you some information about the interaction of residues I and j. Okay, any questions?
Yeah, so this matrix, this is not a distance matrix, but it instead encodes some information about the way they interact. So this is not a distance.
This is not a distance matrix. Those are all abstract, feature spaces, basically. Yeah.
Thanks.
So like 128 numbers which will encode this information in some way. So yeah, basically what I'm saying is that the information flow of the network will be useful for determining which of these proteins should actually be in contact with each other and which should not be. And yeah, I will of course explain this in more detail later. Okay, let's move on. So I will first give like this sort of an overview of the network. So we start with an input sequence. It goes into this matrix at the top, like into the multiple sequence alignment at the very top. And then we run the genetic database search and find evolutionary related sequences and align them and put them into the same matrix. So this is the key input for the network. And then we also create the residue pair information basically just by concatenating the pairs of residues. So nothing fancy happening here. And the final input are the templates. And templates are evolutionary related proteins for which we already know the structure. So we search against the PDB database I have mentioned, and then you can essentially, if it's in PDB, then you know the structure and you can extract that information from there. So, but we also notice that this is not quite as useful for a network as you might think. So it's sometimes useful, but it typically performs very well even without templates. And with these inputs, we go into the actual network. So we have the MSA representation, the multiple sequence alignment, and we have the pair representation. And then we have the Evoformer, which is a key part of the model, which actually extracts the jointly updates the MSA and the pair representation. And the idea being that it extracts the information about what is in contact with which out of MSA and into the pair representation. And there is an information flow back. So you basically can see, I think these two residues are in contact. Let's check whether it makes sense in the MSA that I see. And after that, we have a very highly processed MSA and pair representation. And we use that. So we take basically the first row of the MSA, which corresponds to your initial sequence, and the pair information, and feed it into another neural network, which is called structure module, which predicts the position of each residue in a special parameterization, which I will also talk a little bit about later. And then we predict the chi angles for the side chain. So basically like angles which are sufficient to describe how these side chains hang on the sides. Does the overall construction of this make sense? Yeah, and this is an end-to-end trained network. So you have like gradient flow through this all.
Could you please clarify what exactly do you predict? You mentioned angles. How angles provide this partial structure?
Yes, so I will have a slide about this later, but the idea is that for each residue, like the backbone, it is sufficient to predict a translation and rotation. So yeah, let's just, I will answer about the backbone later, okay? Because I have the slides later. But for the side chains, basically the idea is that you can represent them. So all side chains have like 1 to 4 degrees of freedom, which are angles, but they have like 20 atoms or something. So you don't really need to specify all 20 atoms positions. It is sufficient to just give 4 angles, right? Just because they are quite rigid in most of the degrees of freedom. So we just predict those chi angles for the side chains. Okay, and a very cool trick we have here on top of that is called recycling. So we take the outputs of the network and we feed them back into the network again. So we take this structure and we put it back here. We take the final features and we put them back here. And we ask the same network with the same weights to just try predicting the same protein again. And we do this 3 times. So in total, this becomes like 4 executions of the model. So instead of 48 blocks, you actually have like 128 92. And the key thing here is that so during training, we only backpropagate through the final recycling iteration. So we randomize the number of recyclings. So it can be 0, 1, 2, 3, and we only backpropagate through the final one. So it's like truncated backpropagation, basically. And that allows to save a lot of memory and also to speed the training, because you don't need to backpropagate through all these layers. And actually, I don't know if Dmitry remembers, but this reminds me a lot about of the auto context, which I worked on in my first year of the PhD. So that was, yeah, when I first heard about this idea, I was like, well, this sounds somehow familiar. And then I remembered, yeah, I worked on something related.
Sure, I remember, yeah. And I started thinking exactly about that.
Yeah. So yeah, it is quite a lot like auto context, except reborn in the deep learning times, I guess. Yeah. So that's recycling. Any questions about it? Okay, let's move on. Another outputs of the network are the confidence estimates, and I think this will be really interesting for the Bayesian methods community. Okay, let's just first say a few words about what they are. So one of them is a per residue confidence, which basically says how confident the network is in the prediction in each of each residue. So like it's confident here, but not very confident here. All right. And then another one is the predicted aligned error, which is a pairwise confidence. So that allows you to say whether two residues are located correctly relative to each other. And if you look at these matrices, what you can tell is, let's say you have two blocks of the protein. They're often called domains, like this sort of structurally independent blocks. And they may be in a certain position. And you want to answer the question of, does the network actually place them in that position because it's confident they're in this position, or it's just it didn't have an idea, so it put it in some random orientation relative to each other. And both of these are very useful for interpreting the structures. And biologists definitely take a very close look at those before thinking whether they can trust the prediction. But the mind-blowing thing for me was that you can train these confidence measures just by supervising the network on the training set. So basically, the network predicts some structure, you compare it to the ground truth, and then you can supervise this prediction heads so that they regress basically the some value that you get by comparing the prediction to the ground truth. And so essentially the same thing happens here. And that seems to work quite well. Like they are well calibrated and definitely useful. So yeah, maybe one obvious thing is that this probably means it does not completely overfit on the training set, because otherwise you would basically get just perfect confidence everywhere. But yeah, still, as a person coming from computer vision, this was very surprising to me that you could pull this off. And we also use these confidences for ranking of predictions which come from different networks. So you can train multiple networks, and you can just basically compare their PLDDT to figure out which one of them is best. Any questions? Okay. Now let's dig a little bit deeper into the Evoformer and structure module. So with Evoformer, what you have is the multiple sequence alignment and the pair representation. Okay. And the first interaction you have is between the pairs and the MSA representation. And what is happening here is that you have attention which operates within each row of the MSA independently. So in each row, you have like its own attention operation, like in regular transformers. But the logits, like the attention weights for that, are biased by the pair stack. So the pair stack basically tells you whether, let's say residues I and J should talk to each other or not. So we use it to modulate the attention operation here. And that happens in every row. Make sense?
Yeah, I have a question.
Sure, yeah.
So this representation, the one on the bottom, this corresponds to the first row on the top, right? So we have a human protein and it is on the bottom, but the bottom row biases the attention of other species as well.
Well, basically, we assume or hope that all of these rows share the same structure, or at least it's similar enough, right? Because this is all like neural representations, it can obviously hold more than one possible structure in this pair representation, right? So it shouldn't be a huge problem, but yes, we do bias all of them in the same way, which might be a problem, but does not seem to be, and definitely makes it much more computationally tractable.
Okay, thanks.
Yeah, and then we have another attention operation, which basically updates all columns of this independently. And now we need some sort of information flow back. So the pair needs to be updated given the MSA. And we use an operation which we call outer product. So what happens here is we basically compute the outer product of all positions in this matrix. So like, we take the features for position I, for position J, and we take an outer product of them, right? And then we average that over all sequences. So you basically take an average over rows. And you get something which has the correct dimensions for the pair matrix. So it has like I times J times some channels. And we feed that into the pair channel. Why this makes sense? It's basically a generalized correlation function, right? So you can compute correlations between different positions in the MSA, but it's done in a very abstract space and also after lots of these attention operations. So it can extract very high order operations in reality. High order correlations. Make sense? Okay. And the final update is the triangle interaction on the pair matrix. So basically, this is something I will talk about now, and I think, I think this is very, very cool. So, roughly speaking, you are allowed to the network to figure out whether the pair representation is self-consistent or not. Okay, let's go to the triangle interaction, and just to give you sort of an intuition of why a triangle interaction is important. Let's say we have 3 points, A, B, and C. And the realization here is that if you know the distances of AB and B and BC, then you have a constraint on the AC, right? Like there is a triangle inequality, for example. And from the multiple sequence alignment and the sequence information gives you some relationship between the residues. But then you essentially need to propagate them through your whole pairwise representation. And so given that the pair embedding encodes relations, the insight here is that if we have a pair AC, then it should depend on BC and AB. Okay. Now, if we go into the graph framework, so if you are familiar with graph networks, they will typically talk about like nodes. You have nodes, and they interact with each other, right? And that's basically how you construct the graph network. Here we also have a graph of residues, but we are more interested in talking about the edges of that graph rather than the nodes of the graph. And the edges are basically pairs of residues. And we don't know the graph. We need to infer it. So the graph of spatial closeness of different residues. And the triplet relation in this sort of language are the length 3 cycles in the graph. So we have like, 3 different residues, and we need to consider all these sort of cycles. Now, what this means practically is that if we have this green arrow, we want to update it based on all possible cycles of length 3, which involve this edge, right? So we want to basically iterate through all possible intermediates for that. And this allows us to incorporate this inductive bias of transit, like triangle inequality or loop closures and things like that into the network architecture. Does this make sense?
Yes.
Great, thanks. Yeah, so this is basically how it operates. So it's you can look in the paper into the details of how this is implemented, but this is basically the idea. Like you consider all possible intermediates and you construct some update operation which has this sort of form. And if you look at it, it becomes quite similar to matrix multiplication with some nonlinearities. Okay. Now, let's talk about the structure module, which is what actually creates the structure based on this representation extracted here. Now, the structure module does end-to-end folding. That's maybe an interesting aspect of that compared to the first version of AlphaFold, which used gradient descent. And again, the mind-blowing detail about this is like the very starting point for the structure module. We assume that the protein backbone is a gas of rigid bodies. So what does this mean? If you look at this backbone, like the one we discussed at the very end, this thing, it has very specific geometry. You can fit a rigid frame, like basically this sort of triangle, into each backbone, into each residue's backbone, right? So it basically is defined by the translation and orientation, right? And then we just throw away the chain completely. So there is no notion of the chain in the neural network itself. Like it does not know that it is not forced to make a chain of residues. So you just have this gas. So all of these frames may move independently from, or rigid bodies may move independently from each other. And then they fly around, and due to the pressure of losses, they form a chain. Does this make sense? Yes. So in order to actually construct the to update these rigid bodies, we have a special 3D equivariant transformer architecture, where the key method is called invariant point attention. So basically, it looks like this. So as you go through the iteration, it somehow wiggles around the protein to find sort of the best possible structure for it. And then that gives us the position of the residues, the backbone of the residues. And then, as I already said, we build the side chains using the torsion angles. Or
Michael, I have a question.
The same thing. Yep.
How do you ensure the consistency of the backbone itself if you ignore it?
So there are a few ways. One of them is just the losses from comparing to the PDB structures, the ground truth, because all of them form a chain, right? So even if you do nothing special, like most of the time you will get a nice backbone chain, just because otherwise you can't fit the ground truth very well, right? The second way is we have an explicit violation loss, which penalizes these violations in the backbone. We only use it like in the fine-tuning stages, because otherwise it basically interferes with learning early on. But in the fine-tuning, we have that. And we have the third thing, which is after we have constructed this protein structure, we have a relaxation process. So basically, we take an energy function which ensures that the protein geometry is good, and we minimize our structure with respect to that energy function. And that is like a post-processing step. So we just take the final prediction and ensure that all the geometry is good.
Okay, thank you.
Yeah.
Artem, please turn off your mic. You have strong background noise.
Okay, let's move on. I can't hear Artem anymore, so let's move on. Now, those are the local frames that I've talked about, and maybe it's not completely obvious why local frames are such an interesting thing. And the reason for that is that if the coordinates in this local frame are invariant to the choice of the global frame. So what I mean by that is that, let's say you're this frame, and you want to find the position of this frame here, like basically this point, right? So you can say, okay, it's located at this, like the vector is this, right? So you can see this vector. And now the great thing about this is that if you rotate this whole and translate all this picture, this relative vector will not change. So it stays constant with respect to the choice of the global frame. So this is what gives this the equivariance properties. Right?
Could you please repeat one?
Yeah. Okay, Dmitry, you're breaking up, but yeah, I'll repeat it once again. So basically, the idea is that using these local frames, you can measure coordinates in the local frame, right? Mm-hmm. So imagine that you are you go into the frame which is defined by this triangle, right? So you're facing that way. This residue is to the right, this residue is to the left, and then you say, okay, I want to know the C alpha atom of this residue, and you measure where it is relative to you, okay? Mm-hmm. And the key insight here is that if you would rotate this whole picture somehow, or you would translate it, this coordinate will not change. So the residue to the right will stay in exactly the same position relative to you. So if all you're doing is measuring these local coordinates and producing updates also in the local reference frame, that the whole construction will be invariant to the choice of the global frame. So you don't care about what the global frame was.
Yes, but doesn't it mean that for any frame you have lots of local coordinates?
Yes. That means, well, we assign a local frame to each residue, right? So we have, like, let's say 100 residues. Each one of them will have its own local frame. And this residue will talk to all other residues geometrically only through that local frame. So you will be, yeah.
So that's just unique local frame for each.
Yeah, it is updated between iterations, but when residues are talking to each other, it is fixed. Yes.
So for each residue, there's just unique local frame, right?
Yeah, yeah.
And these unique local frames, they're updated during iterations.
Exactly, that's right.
Mm-hmm.
So you first use them to talk to each other, and then you update them. And we also use the same frames for computing the losses. If you remember, I talked about predicted aligned error, and this alignment basically is also done using these frames. So it's like a thing we carry through the entire project.
Okay. Now it's clear.
Yeah. Okay, great. Any more questions about this? Okay. Oh, there is something in the chat. Looks quite similar to film, but that was probably something before that. Sounds a lot like plane relative attention in sequence transformer. Sorry, Dmitry, do you mean the local frames or something else?
Sorry, I mean local frames. So you just explained that local frames means that each residue is communicating to all the other residues within its own coordinate frame. This sounds a lot like the situation in sequence transformers, where there is relative attention and the positional embeddings for each iteration of the attention are measured relatively to each character. So the signals are sent in the local frame for each character, so to speak.
Yeah, I think there is some similarity. It's obviously quite different because this is talking about like 3D spaces and relative attention is talking about like abstract spaces, right? So there is a very significant difference there. But yeah, the overall spirit of this, I think, is similar. And also, like my intuition on why this helps is maybe not even because of it doesn't matter what global frame you choose, but because you can wiggle different parts of the protein independently, right? So if you have like a block which will move jointly, right, then you, from the perspective of the residues in that block, nothing will change, right? So they will all stay in constant position relative to each other, and they can measure that. So in that sense, it is similar to the relative attention, I think. So that is also why relative attention helps, because if you have, like a sentence, you mostly care about how things within that sentence work. You don't care about the fact that this is like, token number 200, right?
You could say that this is an extension of the one-dimensional attention in sequence transformers to 3 dimensions.
Yeah. Oh, wait. Sorry, could you repeat that?
This sounds like an extension of single dimension attention in sequence transformers to 3 dimensions, where you just can look in 3 dimensions.
With the caveat that you need to choose these frames, right? So you have to somehow assign a frame to each one of those. But yeah, with that caveat, yes, it is. So it is an invariant point attention, and attention means that it also has the like regular attention components, like 1D attention that you typically have. Cool. Now the question is, which parts of this mattered? And for better or for worse, the answer is all of it. So we have performed quite a few ablations for the paper, and we couldn't find like a single improvement which was completely dominant over all others. And we think that this means that the important contribution was the methodology of building the protein intuition into the model, rather than individual bits of like this is like the arrow you really want to have in your network. And another thing is that thing we've noticed is that if we perform multiple ablations, like we remove 2 things at once. So this is IPA is this 3D attention we've talked about, and recycling is also something I've talked about. You get quite a significant drop in performance, and this highlights that there are interactions between the components, and if you remove too many of them, you lose a lot of performance from that. Okay. Now let's talk about how AlphaFold understands protein. And does anyone want like a few minutes break maybe? I think we're, that's all on the like ML heavy part, and I will just essentially talk about how AlphaFold understands protein, show some pictures, things like that.
Okay, well, we're looking forward to see it.
Okay, so no break, we're good to go.
Right.
Okay, let's see. So how does AlphaFold understand protein? One of the things about the computational structure prediction, there is a question about templates. Let's just answer it at the end, just to avoid breaking the flow. So on the biological context, the computational structure prediction, as I have described it, is quite underspecified. So you basically have the sequence, and you want to know the 3D structure of your protein. The problem is it doesn't work that way. There are lots of things which proteins interact with. And when you create the, when you experimentally determine the 3D structure, it's often important to, for example, express the protein with some ligands, like, or just simple atoms like zinc. Or many proteins actually only form folded structure when there are multiple copies of the same chain. So you have, like, in this case, 3 chains, and they come together to create a single protein. There might be DNA which the protein will bind to. There might be experimental conditions, like you could, change the pH of your solution. There might be multiple conformations. Some proteins, like most proteins, probably adopt different states in order to do different things. And we saw that with this pump earlier on. And the network doesn't know any of that. So what happens is that it basically infers the missing context, which is both surprising and not surprising. But you see these things like this, a protein with a zinc binding site where there is this zinc. The blue is the AlphaFold prediction, and the green is the experimental structure. And you see that these side chains just align perfectly to each other, despite the fact that AlphaFold was not told that there is a zinc in there. And yeah, so that's ligands. And then there are multimeric predictions. So this is a cusp protein which is a trimer. So it basically there are 3 copies of the same chain which form a structure together. And if you take an AlphaFold prediction, like just the prediction for a single chain, and you take, if you move them in the right positions, then you get this absolutely perfect prediction for the trimer. So the network definitely knows that it should form a trimer, but it just can only show you one of these 3 copies. Okay, I will also take the question about the uncertainty later. Okay, the question about zinc and the energy function. It's actually a good question. I think it's somehow we're using the Amber function to relax it, and it's using reasonably strong constraints, which I think constraints to the initial solution of AlphaFold. So I think that's why it does not blow it up completely. And another thing is that we mostly minimize the Amber so that we fix up the geometry, and geometry has like really high contribution to the energy, as opposed to the things like which could like if you have 2 positively charged things close by, then probably Amber will be a bit unhappy, but not extremely unhappy, if that makes sense, as opposed to breaking the backbone. Okay, let's move on. Now, we wanted to figure out what is happening inside the network, because it's clearly doing something interesting, and can we see what is actually happening there? And what we did here is we took these Evoformer blocks, and we predicted a separate, we froze that, so it's all absolutely fixed. And then instead of just looking at the structure at the very end, with the structure module, we predicted a separate structure. We used a separate structure model, which is trained for each of these blocks. So basically at each block, there is a structure module which tries to predict the final structure based on the current features. So basically, it's like a linear projection often used in NLP, but it's not linear, but still. So what do we see here? This is one of the most challenging targets in CASP14, which is a SARS-CoV-2 protein called ORF8. We predicted quite well, but we also know that it's a very hard protein. And you see the evolution here of the prediction. The counter goes from 1 to 192, which is like this 48 blocks times 4 recyclings. So you see something which resembles this search a lot, like, it just jumps around, but also once it finds some sort of good configuration, there are these 3 strands at some point they basically latch on, and then it just stops moving them. So it looks a lot like search, at least to me. And this is the largest protein in CASP14, T1044. And you can see that you get some sort of rough guess about its structure quite early on, but then these domains, which are colored by different, I think it's just colored by rainbow. But yeah, anyways, you can see that different parts of the protein just wiggle around relative to each other, right? So it's trying to adjust that quite very, very late into the training, even though the individual parts of the prediction are essentially static. And this is what the quality of the prediction looks like as a function of the number of blocks, Evoformer blocks that have been executed. So the green line is the protein, the COVID SARS-CoV-2 protein. And you can see that it takes a very long time to improve. But maybe surprisingly to me, you don't really see much of the drop, right? Like it basically monotonically goes up. With some jumps, but pretty much all the jumps go up, like very rarely goes down. For some simpler proteins, like this 2 lines on the top, you can see that basically by the 48 block mark, it's already very good and does not really change much after that. So our network is extremely deep, and we think that most of the depth mostly helps for the very hardest proteins. So you probably don't need like this whole depth if you have a very simple protein, but if you have a very complicated one, you might want all of that. Make sense? Okay, let's move on. Now on to the results. So in order to understand how well AlphaFold works, we entered it into the CASP14. And so let me just give you like a 1-minute overview of what CASP is. So it is an assessment of protein structure prediction methods, which is run every 2 years. And it has been run for like more than 20 years now, I think. And it's done in a very, very rigorous way. So the organizers reach out to the experimentalists who are, or maybe vice versa, the experimentalists reach out to the organizers and say like, well, I'm about to find a structure of this protein. Or maybe they already have the coordinates, but they just have not released them. Or maybe they have the crystal, but did not run the diffraction experiments on it. So anyways, they're close to determining the structure. And they tell the organizers the sequence, and the organizers distribute it to the participants. And then the participants have 2 weeks to predict the protein structure, and they send it back. And then the organizers compare it to the actual structure that the experimentalists have found. And this is really the gold standard in assessing this, because as you probably realize, it's very tricky to assess the quality of these methods, and you can have test set contamination, things like that. So this is like really the gold standard for determining whether the method actually works or not. And there was a quite little progress in the CASP performance over the years. And then there was some improvement in CASP 13, where AlphaFold was the best method. But yeah, also other methods were also better than in CASP 12. And then in CASP 14, we saw this huge improvement in the performance from AlphaFold 2. Like it's basically at roughly the 90 GDT mark, which the organizers said is considered to be competitive with the experiment. So it's like they have determined, as far as I understand, this happened quite early on. They basically said like, well, if we're at 90 GDT, that means that you can this is a very useful prediction, and is you can find the same sort of things as you would from an experimental structure. So there is a question on how the progress in the CASP benchmark was not monotonic. So the reason for that is that the difficulty of the CASP competition was increasing over the years, right? So it wasn't extremely fast increase, but nevertheless, you basically would assume that like CASP 12 would be quite a bit more, or CASP 11 would be more complex than CASP 7. Right, so and because of that, even though the GDT maybe was a little bit lower, the methods have definitely improved in that period. Okay, makes sense? And on the bottom right, you see the comparison of the accuracy of the predictions of AlphaFold in blue to the other methods. And this is measuring the median RMSD of the prediction, ignoring some outliers, which like floppy bits of the protein, which typically happen at the tails. And you can see that according to this metric, AlphaFold is like almost 3 times better than the other methods which have entered CASP. So, which is why organizers in the community were so excited about this, because it definitely represented this sort of step change in the accuracy of predictions, and definitely allowed to do quite a bit more things than was possible with protein structure prediction previously.
But what is this
any questions?
Yes, I have a question about this metric. What is RMSD?
So RMSD is root mean square deviation. So you take the predicted structure, you take the ground truth structure, you do the best alignment between the two, and you compute the.
How do you make best alignment between the two? Are you solving a kind of optimization problem?
No, you can do this with
to align two 3D structures together.
Yeah, you just do SVD. You can compute the SVD, which will of a 3x3 matrix, if I remember correctly, which will singular value decomposition. So basically, it's a problem of you want to multiply your. Okay, so first you remove the center of coordinates, right, from both structures, and then you just need to find the best rotation. So it's like a least squares problem.
Okay. Now I got it.
Yeah, and here the 95 basically means that we ignore 5% of outliers.
Yeah. Okay, thanks.
So it's a standard metric, and I think this is the metric which is more interesting in the high quality regime, where you're already quite close. Because it basically requires to get everything right, as opposed to GDT, which is a bit more forgiving if you only get some parts right. Okay, so after realizing how exciting this is and how useful this has the potential to be, we've collaborated with Amble EBI to release an AlphaFold protein structure database, and you can go to this link and see it. So it contains the pre-run predictions for the 21 model organisms, including humans, including mouse, wheat, basically model organisms which are very often used in biological research. And you can see the structures, these confidences, which I have talked about, all there. It is available for unrestricted use, so you can use it for absolutely anything, including commercial use. And we have the plans to expand this to UniRef90. So this is one of these genetic databases, which basically has all the protein sequences which are known to high standard, and that has about 100 million sequences. So we're planning to expand the database to include all of that. Now, what have people been using this AlphaFold structure prediction for? One of the interesting things is that you can use it to improve the experimental structure determination. And you can see this in the case, I think on the right. Yeah, I think on the right. So in this, I think this actually happened during CASP. So someone reached out. Maybe that was organizers themselves. They had some protein which they couldn't find the structure for, and determining protein structure from crystallography data has this chicken and egg problem. You already need to know what you're searching for in order to make sense of the data. So you need to have a pretty good guess, and they couldn't solve it for something like a decade. So it was sitting on their shelf, the data, and they couldn't make sense of it. And just plugging the AlphaFold prediction there immediately gave them the crystallographic structure like for that. So that was extremely cool to see. And yeah, you don't get that sort of impact very often as a machine learner, because normally you just have like a sort of image net or something like that. But here you actually help someone figure out the structure of a protein, which they couldn't do otherwise, despite having the data. And also, it helps to you can use it to make sense of low resolution data, and just to sometimes augment if your experimental structure does not model some part of the protein, you could use it to take that from AlphaFold 2. And this is this paper actually came out just a few days ago, and this is again pretty mind-blowing for me. So in this case, people were trying to make the model of a nuclear pore, which is a very large structure in cells. And they had a low resolution data for that. So very, very low resolution, as you can see here, like this sort of blobs, right? And they have used AlphaFold predictions to actually make like an atomic level structure of that. So they fitted AlphaFold predictions inside this density, which is like low resolution, so there are no details available in the data. And they have constructed this absolutely huge model, which hopefully will be useful for research into how these complexes work. Any questions about this? Okay. Okay, so what's next? When we've released AlphaFold, it was extremely cool to see all the community of people coming around it and just trying to use it for various things. And one of the things people used it for was the prediction of multimers. So as I said, multimers is basically when you have multiple copies of the same protein or different proteins which form a structure together. So like in this case, basically you have like this one chain and this is another chain, but they form a structure together. Same thing here. And people have come up with very cool ways to make AlphaFold predict these multimeric structures, despite not being trained to do that. And yeah, it was really great to see. And some people have even done very rigorous evaluation of that. And they found that AlphaFold 2 was superior to the existing methods for doing multimer prediction, again, without being trained to do that.
Michael, and what is multimer?
So multimer is a protein complex, like this is the quaternary structure which you have alluded to in the beginning, where you have multiple protein chains which form a structure together. So they come together and they form like a multimer, right?
Mm-hmm. I see. So it appears that AlphaFold 2 is actually able to predict this quaternary structure as well, right?
To some extent, yes.
It hasn't been trained. Mm-hmm.
Yeah, which is somewhat both surprising and not as usual, because obviously the interaction between different proteins is the same as the interaction within a single protein, right? In some sense, like the physics is the same, the chemistry is the same, but it's still surprising that this works to the extent it does. So, okay, independently of that, we already had work in progress on predicting multimers, and we basically adapted the inputs, the loss function, and the training procedure so that the network can handle these multimers, and then just trained it all from scratch. And we find that this AlphaFold multimer works better than the solution I have described on the last slide of, well, I didn't really describe it, but basically the hack you can do with AlphaFold to make it predict multimeric structures. And that is, and this class pro is basically what was available before AlphaFold. So you can see that like there is a very significant improvement in performance. And this DocQ score is from 0 to 1, so somewhere around here. So probably there is some room to improvements further, but it's definitely a big progress already. And you can see some examples of the predictions on the right. Okay, So what are the remaining things to be done? And honestly, I think this is not the end. It's more like beginning. I think we will see machine learning being used in structural biology a lot more. And yeah, one of the ways you could use it is expand AlphaFold to other areas of structure prediction. So you could increase the accuracy on the multimers. Using this to predict the interaction between proteins sounds extremely exciting. So which proteins actually interact with each other? So if you would put them in a ball, would they stick to each other? And hopefully you can use things like AlphaFold for that. There is the structure of nucleic acids like DNA and RNA and ligand interactions. So let's say you have a protein which interacts with the DNA, then it's interesting to see how this structure of protein plus DNA would look like, or protein plus some ligand. And then there is the conformational diversity, which is what sort of shapes can this protein take up. Another challenge, which is very exciting, is the structural metaproteome. So the community has basically always assumed that there are small number of available protein structures, like 200,000 or something like that. But there is 100 million genetic sequences, and now we can look up a very high quality protein structure for all of them. And the question is, how do you actually make use of that data? Like, you can't look at all 100 million structures just manually. And we do need new tools to actually make use of this structural data. And the third challenge is the prediction of mutational effects. So when you mutate something in the protein, that changes the structure of the protein and the stability. And AlphaFold is typically not very accurate at that. So it can basically predict some sort of average of basically it's not quite as sensitive to the mutations, which are super important for biology, because, for example, a lot of diseases come from point mutations or just mutations in the proteins. And another thing is the binding affinity. So if you have let's say 2 proteins which stick together, and then you ask, what if I mutate this part of the protein? Will it stick more or less? And that is also very important for understanding disease, as well as just general biology. Okay, and with that, I will go to acknowledgements. Huge thanks to everyone who made AlphaFold possible. As you can see, it's a pretty impressive list of people. And yeah, thanks a lot to you for listening. And with that, let me answer the questions that have been asked during the presentation. Okay, so Boris Yangel is asking, where do templates come into play in the model? Okay, let me just scroll back. So they come into the play here, and basically what happens is you extract the pairwise distances out of this template, and you add them as features, in here in the residue pairs. So it basically tells you, like, these 2 residues might be in contact in this template. And then there is a question from Dmitry. Is it true that you predict uncertainty by training a separate model to predict the error produced by the main model? Okay, that was, where was it? That was here. No, that's not true. We're using the same model, which is, I guess, what makes it amazing, that you can use exactly the same model, just essentially a linear projection somewhere or a small MLP at the end to regress its own confidence. And so it's basically the same features. You just predict both the structure and how confident you are in that. Okay. Question from Anthony on the length of the sequences. The sequences we train on are 256 residues to 384 residues. and we test on up to 2,700 residues. But the average length of a protein is maybe 500 residues. So yeah, basically we can, maybe another thing which I still find very surprising is that you can train on like small proteins and protein crops and extrapolate to this very large proteins at test time. But yeah, the overall this average protein is like 500 residues. Any more questions? Okay, can you talk a bit more about uncertainty prediction? What is predicted variance or entropy? Doesn't it look similar to the standard Gaussian model parameterized by neural network with trainable sigma? So, so what is predicted is there is a metric called LDDT, which is local distance difference test. And it measures the quality of your structure compared to the ground truth. And you can compute it for each residue individually. And that what we do is we discretize that value into 50 bins. So basically, it's a LDDT is from 0 to 100. So you have like one bin for 0 and 1, the next bin for like 2 and 3, etc. And then you just have categorical cross entropy for that. So this is how it's trained. It's basically a categorical discretized value. And for the predicted aligned error, it's also very similar. Like you compute the error, you discretize it into some bins, and you predict that, you have a discrete distribution of the bins. Doesn't it look similar to the standard Gaussian model parameterized by neural network with trainable sigma? Well, yeah, okay, it's not a Gaussian as I explained, but yes, it's definitely just parameterizing a discrete distribution. And I guess the surprising bit is that you would not typically assume that this would work for predicting the confidence of the network itself. Because if the network becomes like overconfident, for example, probably this this confidence estimates will be just wrong. But they seem to be quite well collaborated here.
Okay, any more questions, colleagues? Well, if not, then... А что, собственно, я на английском тогда? Я уж на русский перейду. Если больше вопросов нет, Майк, огромное спасибо за супер клевый доклад. Я прям даже некое чувство гордости испытал за то, что BayesGroup смогла воспитать таких блестящих исследователей. И пусть и косвенно, но поучаствовала в несомненном прорыве, ну уж, я не знаю, цивилизационного масштаба. Я думаю, что благодаря этой технологии можно серьезно с мертвой точки сдвинуть огромное количество прикладных задач, включая там тот же синтез лекарств новых. По-моему, очень клево.
Надеюсь, что действительно будет такой прогресс. И да, спасибо большое, что пригласили. Было очень приятно прочитать доклад про это. И отличные вопросы были. Все здорово. Спасибо большое всем участникам.
Отлично. Все, коллеги, значит, через неделю у нас Владимир Титов, студент физтеха, который, с одной стороны, расскажет, конечно, про гораздо более простые штуки, с другой стороны, как мне кажется, это тоже одна из довольно потенциально прорывных тем и, как мне кажется, шаг в сторону сильного искусственного интеллекта. А именно, как можно объединять текстовые и, соответственно, картинчатые модельки благодаря прорыву в области NLP. И, как мне кажется, с помощью этих технологий, о которых, собственно, Вадим расскажет, можно строить универсальные системы, которые могут решать задачи, которые они не видели на этапе обучения, но которые просто текстом сформулированы, благодаря тому, что они на этапе обучения обучались на одни задачи с текстовым описанием. Теперь мы меняем текстовое описание немножко на другое, но нейросеть способна это тоже делать. Но, во всяком случае, это некая наша сейчас мечта, и мы будем в этом направлении активно работать. Но вот первые результаты по скрещиванию, то есть, грубо говоря, благодаря прорыву в области текстов, можно пытаться использовать текстовое описание как своеобразный мостик, который позволяет нейросети переходить от одних задач компьютерного зрения к другим задачам. И вот первые шаги в этом направлении были в 2021 году сделаны, они как раз Вадим расскажет. Ну а дальше, может быть, через годик расскажем о том, что удалось сделать нам, точнее, удастся сделать.